ions to metal nitrogen isotope ratio. There is no metric effects induced by the presence of carbon and no interferences complicate the analysis. So for the future measurement, more natural sources need to be investigated. For example, the mixtures of nitrate with ammonium sulfate, or the mixtures with nitrate with organic reactive nitrogen. And the application of real aerosols, I collected sample in 2 campaigns. In 2014, one campaign is in Jinan, China, and another campaign is Rome, Italy. So here is the result summary. First, nanosins can be used to separate different nitrogen-containing species which commonly observed in atmospheric aerosols. NO2- second ions are unique for nitrate and nitrite salts, and H4+ second ions are unique to ammonium and amino groups. For cross-mode aerosols, different species can be best separated by negative second ions and positive second ions. For fine-mode aerosols, different species can be separated by negative second ions or positive second ions. Stable nitrogen isotope ratio can be measured precisely using NO2- molecular ions on pure nitrate salts. Long-term measurements on in-house sodium nitrate, the result is 16.9 per mil and 0.6 per mil. We also measure the metric effect between sodium nitrate and potassium nitrate, which is this number. 3, the second ion, 23 sodium carbon 2 can be a serious interference when NO2- molecular ion is used to measure nitrogen isotope composition, especially for sodium and carbon, they present in the same time. 4, the presence of carbon in aerosol-like mixture samples lead to a metric effect in potassium nitrate, which can be quantified by this equation. 5, when Ce- molecular ions are used to determine nitrogen isotope composition, there is no metric effects induced by the presence of carbon, no interferences that can complicate our results. And it's more important, using NO2- as a mass can make these measurements species-specific for nitrate. This is some papers related to this work, and thank you very much.
Yeah, thanks very much. Thank you. Thank you. Start on the right.
Okay. Okay, can you go back to slide number 14? Okay, this is your aerosol generation device.
Yeah.
I believe that the nitrogen tank is clean and clear and stable, but with very sensitive methods, you always have to be worried about contamination.
Yeah.
How about contamination, for example, from exhalation of NH3 by people in the laboratory? We found that being a problem in the wind tunnel lab once. People in the lab exhale NH3, it deposits on the particles, and then you have a contamination. Are these kind of things important for you, or is your system
not so important because we measure NO2- second ions.
Mm-hmm.
So we don't measure positive second ions so far with this equipment.
Mm-hmm.
So I don't know how these NH3 things influence our results.
Okay.
Yeah.
Do you have to worry about contamination when you prepare your aerosol samples from the sampling all the way to the laboratory? Is there problems possibly?
I think that the most serious contaminations is maybe carbon and things, but we measure carbon.
Mm-hmm.
So normally I don't find if we have some contaminations during this whole system, we can get different nitrogen to carbon contamination values. So we can correct them in the future for the calculation system.
Okay, now can you go to slide number 21? Yes, you showed that there is a very clear separation in the various populations of data points.